Everybody have a chance to get dinner before coming? Good, I did too, so that was good. So the exam is what? About 60 hours away?
Oh, God. Please don't.
Actually, I think it's more like about 62, something like that. So. But who's counting, right? How's it coming? Is it good to have it that early or not good?
Yes, you get it done, but you kind of wish you had more time to study.
But now you can focus on your other stuff, though, right? I've been teaching the class for about 10 years, and I've never had one this early. In fact, I've been teaching for about 15 years. I've never, ever had a class with an exam on Monday at 9:30. So you're the lucky ones. I told you guys you were a special class. So it was great singing today. I was very pleased. That was cool. It sounded pretty cool. It's pretty exciting. It's no fun when you're up there singing and nobody else is singing along. So it's good. It's good to hear. You knew the songs, though, right? Yeah.
But like one of the songs I didn't know. So
Yeah. How many had ever heard the Coke song before? Yeah. You had to be as old as I am to understand that Coke song was very popular in the 1970s. And I was just an infant at that time. But all right. So you guys know how these go. I'll handle questions, and then we'll put it out on TV land for everybody. I haven't yet posted the video from earlier today. It's sitting on YouTube right now, but I haven't gotten it prepared for viewing yet. So it should be ready sometime later this evening, I think. So, and then we're done. All right. So what you got? Yes.
So PFK, sorry.
PFK, phosphorylation.
So F26BP activates it, but then it's also regulated by ATP.
Yes.
Both allosterically and by substrate.
So actually, the regulation. That's a good question. So let me just repeat what you said, since people without the microphone wouldn't hear that. She said that she's interested in PFK, and it's regulated allosterically by F26BP and ATP. And she said ATP was also regulated. Regulation was through substrate, is what you said. And that's not completely true. No. So the regulation is actually allosteric. And the substrate regulation, the substrate is just substrate. It's no different than any other substrate. So the key thing in ATP is you get two binding sites. So anyway, I'll let you finish your question.
Okay, well, so how do F26BP, does it just activate it after it's been bound by ATP? Or how does that work?
Okay, so her question is, how does F26BP regulate it? Does it happen just after it's bound ATP or what? That's kind of what the question is, right? Okay, so ATP is a little confusing in this respect, okay? And there's not an order to it, to answer your question briefly. There's not an order. So we could imagine that if we think of order, that if I had F26BP present and it binds to the enzyme, what's going to happen to the enzyme? Good exam question. What's going to happen to the enzyme? And what's the change shape? What kind of shape is it going to change to? It's going to go to R state, right? Because it's activating the enzyme. So the enzyme goes to the R state. What happens in the R state from what we've seen with other enzymes? Binds to substrate better, right? So we can imagine an order with that, okay? The enzyme is odd in that it's regulated allosterically by multiple things. So other things could be playing a role in that activation or inactivation, as the case may be. And like other enzymes, we see that the allosteric activation is not necessary for catalysis either. So the substrate can bind even without that F26BP, which is why I say there's not really an order to it. Does that confuse you even further?
No.
Okay.
So the two binding sites that it has, then one that PFK has, one is the allosteric site for F26BP and the other is for ATP.
So our question is, what's the nature of the two allosteric sites on PFK? I think that's what that is, right? Well, it's actually even more complicated than that, because remember, there's other things that can affect for PFK allosterically, right? So we can see AMP affecting it. We can see I'm not remembering all of them off the top of my head, but there's like 3 or 4 things that can affect that, and they will interact differently with the enzyme. Okay, so AMP, I believe, actually overlaps with the ATP binding site, and because it binds slightly differently, it favors the R state instead of the T state. Okay, because the ATP is favoring the T state. But F26BP has its own binding site. So this enzyme has several places it can bind things, and the effects are different depending upon which one is bound. So I'm not sure if I'm answering your question or not, but that's kind of what's there. Connie?
Can you say the sequential model works for this when it comes to R state and T state?
Can I say the sequential model works for this? No, I can't. Okay, and the reason I can't say the sequential model works is the sequential model relates to the binding of substrates, not of allosteric effectors. So the sequential model has to do with one thing followed by another thing binding, and not different substrates binding. Okay? Yeah, question.
So I know
I'm sorry, back here. I'll come back to you.
So it's still on the PFK regulation. And
Yes.
in your highlights, it said something about the KM, and that kind of confused me. Can you go over what?
Sure. So that's a very good question. It's a very good exam question too. So, in the highlights, I talked about the Km of the binding sites of ATP. Now, it may seem a little bit confusing, and I apologize for that. Technically, Km only relates to the binding of substrate, okay? Because we can measure the Km of the binding of the substrate as part of the Vmax calculation. The Vmax over 2, the substrate concentration that gives us the Km. But if we think of Km in a very broader sense, the way we've used the term Km is to measure the affinity that the enzyme has for the binding of a molecule. Let's not call it a substrate at the moment. Okay? Real good example, how about hemoglobin? It's not a substrate because it's not an enzyme reaction, but we can say that the affinity of oxygen for of hemoglobin for oxygen changes as oxygen concentration changes, right? So we can think of Km as a concept in terms of the affinity that a protein has for binding to another molecule. And that's important because when we talk about the binding of ATP, we have both a substrate binding of ATP and a non-substrate binding of ATP. Okay? So the non-substrate binding of ATP occurs in the allosteric site, and we can imagine that there is an affinity as such for that. Right? How much affinity does that allosteric site have for ATP? And as I've talked about in class, it's important to recognize that that allosteric site on PFK has less affinity for ATP than the active site does, as the substrate binding site. Okay? And that's important because only when the substrate concentration is high do we want that allosteric site bound. When the substrate concentration is low, we want that substrate binding site to be having preference. It's taking that ATP because we want that reaction to go when the ATP concentration is low. So when I talk about two different KMs, that's what I'm referring to. Does that make sense? Yeah.
So does PFK have different, like, I don't know if this is only for enzymes, but does it have different subunits, like the R subunit and the catalytic, like these R and C subunits, or is it just different sites that these bind to?
I'm not sure. Could you repeat the question?
Does PFK have different subunits, like regulatory?
Oh, I see. Does PFK have different? In other words, is PFK like ATCase, where in ATCase we saw regulatory subunits and we saw catalytic subunits. And not all enzymes have that setup. So PFK does not have that setup. But it still works in an R and T state because those bound substrates can, in fact, change the protein shape, and that's what's doing it. So, good question. Yeah. Yeah.
Could you repeat that? Did you say that the ATP has a higher affinity at the Ulster site?
Okay, so I'll repeat. She wanted me to repeat the question, repeat my answer about the affinity of the enzyme for ATP. Okay, so we have two sites to think about. We have an allosteric site, which is a control site, and we have a substrate binding site, which is a catalytic site. They're both on the same protein. It's not separate, okay? When we think about those two sites, the one that we want to have the highest affinity is the substrate binding site, meaning lowest Km is going to be where the substrate is bound, because we really want that enzyme to be able to grab that ATP when necessary. The highest Km is the allosteric site, and that's because we don't want this enzyme binding ATP at that site unless ATP concentration is high. Think about this physiologically. When the ATP concentration is high, we want to turn off glycolysis. We don't want this reaction going. When the ATP concentration is low, we want this reaction going, right? Now, somebody said in an emailed question to me the other day something about, well, low ATP turns the enzyme on. And I said, well, that's not really accurate. Okay? It's one thing to say that high ATP turns the enzyme off, but it doesn't mean that low turns it on. There's nothing to turn on. It's just not turned off. Okay? So that's important to keep in mind. Does that answer your question? Okay. Yes, Connie.
Just to double check, a higher Km value means lower affinity for a molecule?
High Km always means high Km, low affinity, low Km, high affinity. Inverse relationship between the two. How many people have their note cards filled out? How many people have started their note cards? How many people are writing the smallest they've ever written in their lives? Yeah. I love looking at the note cards. I mean, it's incredible how tiny some of the writing gets. It's really awesome. Works of art. Yes?
Are note cards front and back?
They're front and back. They're side if you can write on the edge. Okay?
And then
What they all have to be is they all have to be written in your handwriting. They can't be run through a printer. They can't have something taped onto them like a figure in there. It's all going to be in your handwriting. And you have to put your name on there, too. Okay?
And then we just can write our name like in Sharpie when we're after
Yeah,
the test.
when you're done, write it in Sharpie across the front of it. That's fine. Yeah. Just want to make sure you turn your card in. Yes, sir.
My question has to do with the last reaction of glycolysis.
Yes.
And so the delta G0 prime is very negative for that Big Bang step.
That's correct. So he's talking about the pyruvate kinase reaction with a very large negative delta G0 prime. Yeah.
So when you're talking about the feed-forward activation of fructose 1,6-biphosphate with respect to pyruvate kinase,
Yep.
basically my question is, since the delta G0 prime is already very negative, why is the feed-forward activation necessary?
That's a very good question. It's a very good question. So let me just show you the figure, and then I'll come back and answer your question. I like that question, actually. It's a very good one. If we look at glycolysis, what his question concerns is feed-forward activation. And feed-forward activation is important when we think about where it occurs, where my good figure is. It's up here. I think it's here. Okay, hit the sit. Okay. So what his question concerns is fructose 1,6-bisphosphate activates pyruvate kinase. And pyruvate kinase already has, or catalyzes the reaction, has a large negative delta G0 prime. So his question is, well, why do I even need fructose 1,6-bisphosphate if I've already got a reaction that's very negative like that? Okay. Anybody want to take a stab at it before I do? Hit me. What's that?
Regulation?
It is regulation, but his question is, why do we have to have a positive regulation when we have this going on?
So it doesn't run backwards? Or doesn't go
Well, that actually is important for turning it off, not for turning it on. So
To pull the products from the fructose 1,6-bisphosphate reaction.
it's important for pulling the products, but again, it's already pulling the products. So why do we have to worry about that? Well, okay, so it's important for pulling for the aldolase reaction, but his question is, I mean, it's a very good question. His question is, this reaction is already pulling. It's already pulling all that stuff. So why do it more? There's something else about pyruvate kinase. Liz?
Maybe this is more similar to what I said before, which is, isn't that delta G really positive for the F1,6 bisphosphate?
For the aldolase reaction you're talking about?
Yeah.
Yeah, so the delta G prime for the aldolase reaction is very positive. But that's not his question. His question is, why do we have to activate this enzyme?
You have like this really big buildup of F1,6 bisphosphate in there because it can't get over some of delta G prime.
Well, that's what it's trying to do. It's trying to get over the aldolase hump. But that's not the question. The question is, you've already got a reaction that's very favorable. Why do it? Yeah.
If you have the F1,6 present, if you're going through glycolysis
Uh-huh.
rather than gluconeogenesis, so
Well, no, it's not. It's not because you're trying to make glucose. No. Yeah.
Is it because on the other negative, delta G negative reactions, don't they use ATP? And on this one, ATP negatively affects it?
No, because this one isn't affected by ATP. So you guys are all focusing up here. You're not focusing down here. This is where his question's at. All right, I'll tell you the answer. All right. Are there other things that are regulating this enzyme? Yes, there are. Are there negative regulators of this enzyme? Will they turn this enzyme off?
Yes.
Okay. So as the F16BP concentration increases, what's the likelihood it's going to bind to a positive activator instead of a negative activator? What's the other regulation of this enzyme? Phosphorylation. Phosphorylation made it less active. This could help to make it more active. Okay? So it's a very, very good question. I've never heard that question before, but it's a very good question why that's necessary. So the answer is it's not rooted solely in the delta G0 prime, because obviously the allosteric effector is not affecting the delta G0 prime. It's affecting what percentage of the enzyme is active. Make sense? Good question. I should have asked that on the exam. I would have stumped the whole class.
Repeat that answer?
Repeat the answer?
Yeah.
Well, the answer is that there's other things that can inactivate the enzyme. The more F16BP we have present, the more likely it's going to bind instead of the negative things.
They bind at the same spot, I think, like that?
Well, think about what they do. They try to convert enzyme to T, or we convert enzyme to R. They don't have to bind to the same site, but once they convert it to T, is the enzyme going to be very active?
No.
If the enzyme is bound to F16BP, is it going to be very active?
Yes.
Okay, so a greater percentage of the enzyme is going to be active.
Okay.
So it's really just a probability.
It's a probability, exactly. Yes?
What effect does ATP have on pyruvate kinase?
What effect does ATP have on pyruvate kinase? Well, as you can see right here, it has a negative effect. And that sort of makes sense. If we think about, we don't want this reaction going forward any more than necessary if we have plenty of energy. Keep in mind that this pathway is only generating how many net ATPs? 2, right? So most of the ATP that's coming from glucose is not coming actually from the pathway. It's coming from whatever happens to this. We'll talk about this next term. So next term, this guy is going to go to acetyl-CoA. Acetyl-CoA is going to get oxidized to CO2, and that's going to generate a ton of ATP. So if we can prevent the formation of this, okay, we stop that glut of ATP, because cells only have a certain amount of ATP they can make. Well, there's another corollary to this. So if ATP levels are high, and we start making this, I talked about in class what the consequences of that are. Anybody remember what they are? It goes to fat. Because we make acetyl-CoA, we can't take that acetyl-CoA any further. And the body says, oh, we got plenty of ATP, let's turn it into something we can store. Fat. That's the fructose business, that's right. That's why high fructose corn syrup is a problem. Anybody remember that? Kevin Ahern's pet theory of why everybody's getting fat.
Why everybody's grossly overweight.
Gaining weight. I didn't say grossly overweight. I hope everybody isn't grossly overweight, okay? So I hope I'm not. Yes?
Just curious, is there any way stoichiometrically to define how much extra fat should be produced from bystepping glycolysis and going with the high fructose corn syrup? So maybe you can say like X, like there's this many more molecules.
You can't really do that. Okay, you'd like to. Are you a chemistry major by any chance?
No.
Okay, it sounds like a chemistry question. That's why I asked. You would like to do that, okay? But you've got to realize that we're focusing on one thing and tracing this one thing all the way through. But the cell's needs are diverse, and pyruvate can go to many things, for example. So even though, could I trace it all the way to how many fat molecules if this only went to acetyl-CoA and only did that? Yes, I could do that. But the reality is in the mix of the cell, there's no way that only gets focused into one thing. Yep. Yeah.
How is glucose 6-phosphatase regulated in gluconeogenesis?
How is glucose 6-phosphatase regulated in gluconeogenesis? I haven't told you. Would you like?
Isn't that one of the 4 regulatory?
It is. Would you like me to tell you?
No.
If I tell you and you have to know, do you still want me to tell you? Its regulation, as far as I know, is not allosteric, and it's regulated in a manner similar to hexokinase, which is a substrate level that's a little odd. So we don't worry about it much. I've tried to keep it as simple as I could for you. Yeah.
Can we talk about alcohol dehydrogenase and the steps? Like, I know you're talking about in class how that one we have that one enzyme and we can get a hangover from having that enzyme.
Are you going out drinking tonight, or is that the?
Of course.
Or maybe Monday, right? So, okay. So her question had to do with, could I talk about alcohol dehydrogenase and the how we lack that step, et cetera. So yes, I'd be happy to do that. That actually relates to the 3 fates of pyruvate, and that is actually right here. And let's see, there's a better figure here. Okay. So we'll actually talk about this reaction a little bit next term also, but. Before I talk about this reaction, let me back up just a little bit and give it a little perspective. So the perspective, the reason that this reaction is important, as I noted in class earlier, are because cells have to do redox balancing. Okay? So redox balancing is a very, very important thing. Here's glycolysis from glucose down to pyruvate. All right? And a critical step in that process is that single oxidation. There's only one oxidation step that occurs in glycolysis. It occurs twice because we have 2 3-carbon molecules, but it's the same reaction. That oxidation step requires NAD, and NAD is a limiting substance in the cell. So because there's a limiting amount of NAD in the cell, we have to recycle whatever we convert into NADH. Now, if we have plenty of oxygen present, and we'll talk a lot about this at the beginning of next term, if we have plenty of oxygen present, NADH dumps its electrons into the electron transport system and regenerates NAD, and we're right back here. So as long as we have plenty of oxygen present, we have plenty of NAD present. When we start running out of oxygen, we start running out of NAD. And then fermentation reactions become important. I noted there were two sets of fermentation reactions that occur. One that occurs in animals and one that occurs in yeast and bacteria. So the one that occurs in yeast and bacteria is the one that's depicted on the screen here, and that is the NADH, which is produced in this reaction, is converted back to NAD by this reaction. And I'll show you that reaction in just a second. But this reaction right here regenerates NAD, which can now be used to keep this process going. Well, the more we keep turning this cycle, the more ethanol we produce. And as someone in class noted, ethanol is a little toxic for cells, kind of like glucose is toxic for cells. You can't get cells to make too much ethanol because they die. So if you're fermenting, making beer or something like that, you get about 12% or so ethanol, like wine or something. And the yeasts pretty much poop out. They just can't do much more than that. Okay, so if you want to have liquor or something like that, you have to distill it, meaning you get rid of the water and concentrate the alcohol. But to answer your question relating to the hangovers, that actually comes from the next figure, which is this guy right here, which is showing you in more detail that reaction that's going from pyruvate over to ethanol. Right? This, what I'm going to tell you right now, you don't need to know. I'll talk about it next term, but it's a kind of a cool thing. This reaction right here, pyruvate to acetaldehyde, is a very unusual reaction in that it's a non-oxidative decarboxylation. Okay? It's a non-oxidative decarboxylation. We lack the ability to do that. In our cells, we combine the oxidation and the decarboxylation in the same enzyme. Okay? So we can't go to this intermediate from pyruvate. Bacteria and yeast can do that. And so when they get to this intermediate, then it's a single step for them to convert their NADH back to NAD. Now, what I said in class was, we've got this enzyme. We can't go here, but we can certainly catalyze this reaction. Of course, this reaction in us is detoxifying ethanol and making something else that's probably fairly toxic, but that gives us the hangover, and that's acetaldehyde. So when we go out and we have a lot of alcohol, our liver converts this back. My thing is beeping at me. I think my battery is dying in this. It is, okay. Our liver converts ethanol back to this guy, and it's this guy that gives the hangover.
So you can't get rid of it.
Yeah, well, again, about all we can do is either go here. It turns out there's some chemistry here. So what's acetaldehyde? Why does acetaldehyde make us kind of sick? Anybody know? Think about chemically. What's its nature? Come on, organic chemists. What is this?
Wild stab in the dark, is it like a nucleophile of some sort?
Is it a nucleophile of some sort? Well, that's not what I'm looking for. Chemically, what kind of molecule is it? An aldehyde. Its name tells you, right? And what do we know about aldehydes? They're not reduced. This is a reduction, okay? Chemically, they're very unstable. They're very readily oxidized. So what happens when you oxidize acetaldehyde? Is that? No. Come on, folks. Oxidation. We go from an aldehyde. What does an aldehyde get oxidized to? An acid. We get acetic acid out of this. Okay? So you're making vinegar. And that doesn't even take an enzyme. Acetaldehyde is very reactive. And so once you start making up much acetaldehyde, it rapidly goes to acetic acid. And that's probably what actually makes you ill. Go drink a gallon of acetic acid and see if you have a hangover. That's Kevin Ahern's pet theory about hangovers, by the way. So it kind of goes along with Kevin Ahern's pet theory about why we get fat. Yes, ma'am?
So you said that NAD is a limiting resource in the body. Does that mean we just can't make any more? Like, where does NAD come from originally?
Yeah, it's a good question. So NAD is a limiting resource in the body. Does that mean that, does it mean we can't make any more or whatever? It means that cells are very efficient things, and in general, cells never make more of things than what they need. And so NAD is one of those things that it makes a certain amount of, and that's pretty much what a cell has because these processes work so well. Yeah. If it didn't, then cells, I think, would make a greater stockpile of them. But because they've got these backup mechanisms like fermentation and so forth to recycle, they don't need to make more than that. And whenever cells can save energy by not making too much of something, they're better off because they can use that energy for something else. I'm going to challenge you guys with something here. I've never had a class do really well on 2 straight exams. Never. Okay? If you guys do, I don't know, I'd go dancing down the street or something. So,
with all blue hair.
What's that?
With all blue hair.
With all blue hair. Okay. Seriously, I'd be very happy if you guys really nail this exam. So let's see you do it. Yes.
When G6P, it can go into 3 different
Yes.
places.
Yep.
Did you? I don't know if you said or not what NHBAP or NHB.
pH.
I wrote it just now.
Okay. Yep.
Do we need to know where that goes and what that affects?
So I'll just say a word here. So her question has to do with glucose 6-phosphate, which I showed a figure that shows it can go 3 directions. That was actually in the glycogen metabolism. So let me show you that real briefly. And fates of G6P. Anybody see it? Yeah, okay, there we go. Yeah. So glucose 6-phosphate, all right? So glucose 6-phosphate can go to pyruvate. That would happen through glycolysis. Glucose 6-phosphate can go to glucose. That would happen by gluconeogenesis. And it can go to NADPH and ribose by the pentose phosphate pathway. So we'll briefly talk next term about the pentose phosphate pathway. No, you're not responsible for it here. And you're not even responsible for knowing this. But briefly, I'll tell you, ribose, of course, is a sugar that's needed for making nucleotides. So we'll talk about that a little bit then. And NADPH is related to NADH. NADPH is used mostly in anabolic reactions, making things. So we'll see when we're making fatty acids, for example, NADPH is needed. So cells tend to be kind of biased. They use NAD in oxidation reactions, and they use NADPH in anabolic reactions. So they have to have a source of that, and this turns out that glucose 6-phosphate gets oxidized in this pathway to make NADPH. Yes, Connie.
Question about the structure of pyruvate carboxylase.
Pyruvate carboxylase structure question.
You said it has biotin.
Pyruvate carboxylase has biotin. That's correct.
Okay. Where does the carbon that the carboxylase use attach to the biotin?
Does the carbon that the carboxylase uses attach to the biotin? You mean the carbon dioxide?
Yes.
Yeah, so the carbon dioxide, the reason biotin is used in carboxylation reactions is because biotin actually has very good affinity for carbon dioxide. So we almost always see it associated with a carboxylase. If I can find the figure again, I will show you. That is up here. Let's see. No, I'm sorry, it's down here. Here it is, biotin binding. All right. So biotin is a fairly straightforward molecule that you can see there. It attaches to the lysine side chain on the pyruvate carboxylase. And I thought I had one showing the binding of carbon dioxide also. Yeah. So there's the carbon dioxide bound to it right there. Other questions? Yes?
The F260P activation, you say you're going to ask insulin activates the phosphorylation by PP1 to go to the left side of the reaction, which it shows glycolysis is active on that side.
Okay.
Can you explain why it's insulin that activates that?
Yeah, it's a real. Yes, that's the first time I've been asked that question this term. So I will answer that question for you here. Let me show you the figure that she's referring to, which is this figure right here. Okay. This is the most hated figure of the whole term. Would you guys agree with that or not?
Yeah. I think there was one that was even more complicated.
You had one you hated more than
I
this?
think so.
Okay. Well, this usually ranks pretty high anyway in the hate department. So there's something that's very confusing about this. And what her question is back here is frequently confusing to people. So her question is this. Look at what's happening here. So if I have insulin, I said insulin was activating phosphoprotein phosphatase. And phosphoprotein phosphatase was favoring PFK2. And PFK2 was favoring fructose 2,6-bisphosphate. And that was favoring which glycolysis or gluconeogenesis? What's that? It favors glycolysis, right? Now, it seems a little odd, okay? Because insulin is favoring glycolysis. We've already eaten a big meal, and now we're breaking this thing down. Why do we want to do that? Because glucose is a poison. So now the reason she's confused, and the reason it's a very good question is, let's think about what insulin's doing. Insulin is stimulating the synthesis of glycogen. That's an anabolic process, right? And it's stimulating glycolysis, which is a catabolic process. Right? You probably haven't thought about that before. Why is that a problem? Well, it turns out it's not a problem. It's only if it's in the same cycle. Right? The reason that both of these are being stimulated is they both act to reduce the concentration of glucose, and it's what you said here. Glucose is a poison. If you put it into glycogen, it reduces the concentration of glucose. If you break it down in glycolysis, it reduces the concentration of glucose. That poison concentration is reduced. Does that make sense?
So does it matter when each one is used? Really?
Does it matter when each one is used, meaning?
Like when is glycogen formed and when is glucose broken down?
When is glycogen formed and when is glucose broken down? Simultaneously.
Oh, okay.
Yeah. Yep. And the opposite effect, let's go back to the opposite effect. We do epinephrine, what's going to happen? Epinephrine's going to favor gluconeogenesis, which is an anabolic process, and it's going to favor glycogen breakdown, which is a catabolic process. Why does the cell do that? To get glucose out into the bloodstream. Both of them combine to make glucose out of the bloodstream. I mean, this system is pretty beautiful, I think.
So if you use gluconeogenesis and what?
So
epinephrine?
Epinephrine favors gluconeogenesis and glycogen breakdown. Both of those allow the liver, for example, to export glucose. Yes, I'm sorry.
And glucagon was the other one, right?
Glucagon acts very much like epinephrine does, but glucagon only acts in the liver. Yeah. Yes?
Does this also occur in muscle cells?
This process also occurs in muscle cells.
I remember when I was rewatching the video and thinking about it didn't make sense for it to occur in muscle cells.
That's a good question, Connie. I don't think it's an important pathway in muscle cells. It's a very important pathway in liver cells because of the liver's role in modulating glucose. I don't think that it's a very. It's a good question. I can't tell off the top of my head, but I don't think it's a very important pathway in muscle cells. No. The needs of muscles are very different than what the liver's doing. Information overload? Yeah. Connie?
Sorry, quick question about that enzyme that's named PFK2 and FBPase2. Does it have a name that incorporates both those two domains?
Charlotte?
Okay.
What's that? Let's see. We give the name of Lucy. What do we call Lucy? Oh, that was the UDP glucose pyrophosphorylase. That's right. Yeah. I guess the way a lot of people refer to it is by whichever one happens to be active at the time.
Okay.
So when this is active, you call it PFK2, and when this one's active, you call it FBPase2. Yeah. It's confusing. It's a cool enzyme, though. I don't know of any other enzyme in metabolism that's like this one. Yes?
Sorry, can you define a reciprocal regulation?
Can I define a reciprocal regulation? Certainly. Reciprocal regulation occurs when something, that something can be a molecule, that something can be a modification, or whatever. Modification being, say, a phosphorylation, dephosphorylation, for example. Reciprocal regulation occurs when something has opposite effects on catabolic and anabolic pathways. That is the same ones, like glycolysis and gluconeogenesis. So when we look at a prime example of a reciprocal regulator, F26BP, is an excellent reciprocal regulator because it favors glycolysis and it inhibits gluconeogenesis. An example of a reciprocal regulation involving covalent modification is that of glycogen phosphorylase and glycogen synthase. Phosphorylation of each of those converts glycogen phosphorylase into the more active form and converts glycogen synthase into the less active form. So I on a previous video, gave my phone number out and said, call me over the weekend. You can do that. And people say, why do you do that? And in a class this size, I get a few calls. I get a handful of calls. Not too many people call. But I've discovered I can't do that anymore because my videos are out there on YouTube. And I've actually gotten a few weird calls from people who have watched the videos on YouTube in various places. So if you want to call me, send me an email, and I'll send you my cell phone number. You can call me. Yeah. Isn't it weird? Yeah, Liz?
Can you talk a little bit about the different types of phosphorylation, like substrate level versus oxidative?
Oh, okay. So the question is, can I talk about the different types of phosphorylation? There are three. Okay. Three types of phosphorylation that occurs in cells. So let me give, the only one you've seen so far is what's called substrate level. So a prime example of substrate level phosphorylation happens in glycolysis. And if we go back to here, that's not a very good figure. If we go to reaction here, okay. This is called a substrate level phosphorylation. The substrate level phosphorylation means that a high energy molecule is transferring a phosphate directly to ADP to make ATP. This guy's full of energy. It's absolutely bursting with energy. Transfers phosphate because it has more energy than ADP does, transfers it to phosphate, and we make ATP in the process. So that's called substrate level phosphorylation. And as I said in class, it's a relatively minor source of ATP for us. Relatively minor source of ATP. Okay? The second type of phosphorylation that we have in our cells is called oxidative phosphorylation. And it occurs in the mitochondria in conjunction with the electron transport system. We'll talk a lot about that next term. Okay? And that one probably accounts in our cells. I'm not a stoichiometrist, so I can't tell you that. But I would wager we're talking about at least 95% of the ATP in our cells coming from oxidative phosphorylation, and maybe more than that. The third type of phosphorylation, and by the way, these phosphorylations all are involving the synthesis of triphosphates. Okay? So these are phosphorylations making triphosphates, making ATP. The third type of phosphorylation that occurs is called photophosphorylation. That's basically what's happening in photosynthesis, where the energy of light is being captured, and the energy that's captured is transferred ultimately to make ATP. We unfortunately don't talk about photosynthesis in this class. It's really unfortunate. But what you learn when you examine photosynthesis is that the electron transport system is very, very, very similar to the chloroplast, what happens in the chloroplast. And so if you learn electron transport, you have a pretty good idea of what's happening in photosynthesis. So those are the three types of phosphorylations that make ATP. Yes, sir? What is what? Why is glucose poisonous? Glucose messes with the osmotic balance of the cell. And so if you mess with the osmotic balance of the cell, you burst cells. That's why. We'll talk next term, again, everything's next term, but next term we'll talk about the need to balance the osmotic balance of the cell, not so much with respect to glucose, but with respect to ions like sodium and potassium. Cells are in a constant battle to balance their osmotic pressures because the membrane that we have is not very strong, and if that pressure gets too great, they burst very readily. So cells have to continuously burn ATP to balance those pressures. Okay. Yeah?
When you were talking about hypoxia induction factor, you gave like a chart that had the amount of enzymes it stimulated.
Yeah.
Do we need to memorize that?
Do we need to memorize the chart that had the enzymes from hypoxia induction factor?
No.
Well, she's declared that you don't. So.
Okay.
Well, in general, what's the rule I have in the class?
Don't need to memorize things unless specifically say it. Sure. I
Yeah.
don't know. You mentioned it in class, though, and you also gave the.
So, in general, if I mention it, it's fair game.
Right.
Yeah.
So you didn't mention it. So.
That's fair game.
Okay.
Yeah. I'll be honest with you. I mean, to answer your question, am I going to ask you to list 6 enzymes that are induced by that? Probably not.
Okay.
But it's something related to that is fair game. So I think you should know what's there. Yeah.
Could you just go over hydrolysis real quick and talk about how specifically hydrolysis occurs? Because water is pretty abundant in the cell, and how it specifically gets targeted on one side.
How does hydrolysis happen? Well,
Just how it gets specific. How come hydrolysis isn't happening on everything isn't getting cleaved left and right all the time?
okay. That's actually a bigger question. We haven't talked about that, but let me just try to address that. If we look at proteins, for example, okay, and we say, well, we've studied enzymes, and the enzymes that break peptide bonds are catalyzing hydrolysis reactions. So why aren't those hydrolysis reactions occurring so rapidly in the absence of that enzyme is part of what you're asking, I think. Okay? And the answer is that you can hydrolyze proteins, and you can hydrolyze nucleic acids. It's easier to hydrolyze proteins than it is nucleic acids. But The answer to your question is if you want to hydrolyze proteins, it takes a fairly strong acid, it takes a strong proton concentration to break that bond. And in the cell, we don't have that happening. We don't have those high proton concentrations. So in the environment of the cell where the pH is relatively low, the peptide bond, and for that matter, the phosphodiester bond of nucleic acids are both quite stable. All right. So that's probably not as satisfying an answer as you want. But if you think about what has to happen in a serine protease to basically hydrolyze a peptide bond, we have to create a nucleophile in order for that to happen. And creating that nucleophile is what facilitates that overall process occurring. So again, as long as we don't have those nucleophiles floating around in the context of the cell, we have much more stability for the bonds. You sure you're not a chemist? Connie?
Sorry, can you talk about hypoxia?
Can I talk about hypoxia? Okay. So hypoxia is a condition where cells run out of oxygen. Cells run out of oxygen. So when cells run out of oxygen, and again, I think that's on here somewhere. Is it up? Other consideration. There it is. There we go. Yeah. Okay. When do cells run out of oxygen? Well, cells running out of oxygen, it's a fairly normal sort of thing. We think about, well, here's this muscle cell, it's exercising, it's running out of oxygen. So cells have to be able to have conditions or be able to handle conditions where their oxygen concentration is low. Maybe I'm a rapidly growing cell. Maybe I am a cell that is in a child, and this child is going through a growth spurt. All right? Well, before part of that growth spurt, not everything gets coordinated real well. So I might have, for example, muscle cells that don't have a real good supply of blood vessels and so forth. And so they don't have as good of a supply of oxygen as they otherwise would need. Yet the rest of the body is saying, hey, we're growing, we got growth hormone, we're going to go do our thing. The body has to have a way of adapting when oxygen concentrations are low that allow those cells to survive. So cells have built into them this ability to handle what's called hypoxia, low oxygen concentration. That hypoxia is a condition, that is low oxygen concentration, and it causes activation of this protein called HIF, hypoxia induction factor or hypoxia inducing factor, either one's equivalent as far as I'm concerned. That is a transcription factor. Now somebody in the class sent me a message saying, HIF makes gluts. It doesn't make gluts. Okay? HIF is a transcription factor, which means it stimulates the transcription of the genes that make gluts. So HIF doesn't make gluts. It's very important to be precise in your language. HIF does not make gluts. It stimulates transcription of the genes that code for gluts, basically. Okay? Why is that important? Well, gluts, of course, are glucose transport proteins. They move to the surface of the cell membrane. And the more of those you have on the surface of the cell, the more likely they are going to be able to bring glucose in. When oxygen concentration is low, it takes more glucose to get the same ATP. It takes more glucose to get the same ATP. If we don't have oxygen in the cell and all we can do is ferment, we get a total of 2 ATPs per glucose. If we have oxygen available, from glucose we get 38 ATPs. So if we want to get the same amount of ATP in the absence of oxygen as we do in the presence of oxygen, we need 19 times more glucose. Well, having more gluts out there makes a lot of sense. You put more things out there that can suck more glucose out of the bloodstream, the cell's going to be more likely to survive. Well, let's think about this. You bring in 19 times as much glucose, but let's say you've got a limiting amount of glycolysis enzymes, right? They're already saturated. They're already at their Vmax, right? So in addition to making gluts, wouldn't you want to make more enzymes of the glycolysis cycle so that you could handle all that glucose? And that's the other thing that HIF induces. It induces the production of many of the glycolysis enzymes. Okay? that allows the cell to do the two things that it needs. Well, that's great if you're a rapidly growing child and you've got these muscle cells that need all this ability to handle low oxygen concentrations. But those same sorts of things are built into tumor cells because tumor cells come from our regular cells. So tumor cells that have low oxygen concentrations will do like any other cell does. They'll start making HIF, and HIF will start making more gluts so they can get more glucose. They will make more glycolysis enzymes. And in addition, they will actually stimulate the growth. They will stimulate release factors that stimulate the growth of blood vessels. And all those combine to successfully allow a tumor to grow. Not all tumors are successful. The ones that we see and we know as tumors are those that are successful. If they, never can grow blood vessels, if they can't handle hypoxia, if they can't, do other basic processes that are important, they never become a tumor. They just die, and the body gets rid of them. Does that help?
Yeah, but I just have another question. You mentioned in class that the tumor cells will activate HIF when it's low in oxygen.
Mm-hmm.
But you said it's low in oxygen because there's no blood vessels.
Mm-hmm.
Then,
Yeah.
But how does activating HIF help if there's also no sugars because there's no blood vessels?
Well, keep in mind, the cells of your body are bathed in fluid.
Okay.
They are bathed in fluid. So even though you don't have blood vessels right here, you've got blood vessels out here.
Okay.
Right? And so everything that those cells are getting is coming through whatever fluid is in the spaces between cells, and that's ultimately coming from blood vessels.
Okay.
So it's just like putting a big suction device here. No, seriously. I mean, that's what it's doing, is it's just sucking more of that stuff into there because it has the additional gluts to make that possible. Okay? Yeah?
So is HIF,
HIF.
does it bind to an enhancer and it's like an activator?
So does HIF bind to an enhancer and it's an activator? And I believe it is, yes. Okay. We'll talk about enhancers next term. But yeah. Yes?
Does it make more NAD as well then, along with other?
Does it make any more NAD as well? No. No. If it did, it might be able to handle things a little bit better. Yeah. Yes? The Boring effect? Yeah, the Bohr effect. So the Bohr effect comes in several ways. That song today didn't tell you enough about the Bohr effect, is that? Okay. Not good enough, all right. So the Bohr effect, way back here, relating to hemoglobin, okay, arises as a result of several things, okay? So let's start with this guy, okay? So the first observation of the Bohr effect was what you see on the screen. That is, if you take a sample of blood and you measure its oxygen carrying capacity at different pHs, within physiological range, of course, that when you measure at those pHs, the higher pH will have a higher affinity for oxygen than will the lower pH. That means that when hemoglobin is found in a lower pH in the physiological range, that it tends to give up oxygen. Well, that turns out to make a lot of sense because rapidly metabolizing cells are producing protons, and protons are lowering the pH. Bang, okay? Cells that are rapidly metabolizing need more oxygen. Hemoglobin says, I'm in a place where the pH is low, I dump oxygen, cells are happy. That's the first component of the Bohr effect. The second component of the Bohr effect is the observation that carbon dioxide also affects that process. Okay? So here was this original condition where we measured pH 7.4, no CO2. If we measure the oxygen binding at pH 7.2 with no CO2, that's the other curve that you saw there. But if we take the same thing now at 7.2 and we add CO2, we see even more dumping of oxygen. Well, that again makes more sense, makes a lot of sense because rapidly metabolizing cells, in addition to making protons, are generating carbon dioxide. So they're dumping carbon dioxide out into the bloodstream. Again, this is a sign that hemoglobin picks up and recognizes that this is a place to dump oxygen, and that's basically what it does. So those two things together make possible the dumping of oxygen at places where it's needed. Now, the other side of the Bohr effect has to do with the transport of carbon dioxide and ultimately protons back into the lungs. So I talked about how carbon dioxide is bound to make carbamates. It binds to those amine groups and makes a covalent bond and gets back to the lungs. The protons can go on to histidines. In some cases, they can go on to other amines as well and get carried back to the lungs. In the lungs, it's a very different environment than was out in the tissues that were being metabolized. In the lungs, the oxygen concentration is excruciatingly high. And though I didn't really talk much about it this term, in the past when I've talked about this in the class, I've made the point that the oxygen concentration in the lungs is so high that it literally forces its way onto the hemoglobin. And in forcing its way onto the hemoglobin, it favors structural changes that favor the release of carbon dioxide. So because of that, carbon dioxide gets carried to the lungs, but it doesn't go any further. Structural changes happen, carbon dioxide is released, and we exhale it. So that's basically the Bohr effect at the molecular level. A lot of people talk about 2,3-BPG as part of the Bohr effect, but it's not. It's a separate phenomenon. Yeah, tell him. About what?
About blood clotting.
Blood clotting. Are you in favor of or are you opposed to that?
Pardon me?
Go ahead.
So in a soft clot, you have control thrombin going to thrombin.
Uh-huh.
And then thrombin can activate hydrogen to 5
Right?
So would your hemophiliac not have thrombin, or do they not have
Okay.
hydrogen?
So her question has to do with blood clotting. So let me go to the figure that I show you for blood clotting and answer her question. This relates to, it's not enzymes, it's catalytic strategies, I believe. And nope, it's not there. It's the next one. Allosteric regulation. Okay. So if we look at the Cascade scheme right here. Okay. Her question is, does the hemophiliac simply lack prothrombin, or does it lack fibrinogen, or what? Well, we could imagine that if a person lacked fibrinogen completely, that they'd be in pretty deep doo-doo, right? Okay. And we could imagine that they'd be in pretty deep doo-doo if they lack this. So people, and there's different types of hemophilia. The people who have hemophilia tend to lack, I want to say it's this guy up here and this guy up here, or this guy up here. So they're further up in the signaling scheme, not down here in the clotting scheme. Because remember, if we get too much or too little clotting, we bleed to death internally. And so probably have some of these things down here, but they're lacking up in here.
Does that mean it takes longer for a blood clot to form?
It will take longer, and it may not form, in some cases, very readily at all. Yeah. So that's why the addition of these factors to the people's blood of hemophiliacs allows them to have a much more normal clotting. You guys are looking tired. Is it time for some acetaldehyde?
Yes. How's that going to help? I have a really silly question.
Connie has a silly question.
For, is it glycogen phosphorylase?
Okay.
For B, you have ATP and G6P that can help convert it into the T state. And I remember looking at the figure, and it could attach in two places.
Okay.
Both ATP attaches, like, there's two ATPs that attach.
Mhm.
There's two G6Ps that can attach. Can it, like, mix and match in any way, shape, or form? Like,
Mix and match what?
Like, attach one ATP and attach one G6P, and it converts that into a T state.
You're right. That's a silly question. No, I'm sorry. I expected a big laugh out of that. So, her question is that we've got a dimer, and can we get one of them phosphorylated and the other one not phosphorylated and then allosterically regulated? And you could imagine a variety of schemes, but that would probably be a question that if I asked you something like that on the exam, I would be crucified for. So I am not fond of crucifixion. Personally. It's my personal dispreference, yes. Yes? Can I go over Western blotting? Okay. Western blotting, going way back. Oops, sorry. Okay, Western blotting, we talked about methods of characterizing proteins. And Western blotting is a technique, and where do I have it? It's up here. Down here? Oh, I mean logical techniques. Okay, Western blotting. Okay. Western blotting is a technique that uses antibodies to basically flag a protein of interest in a mixture of proteins. Okay? Well, imagine, if you will, that I had isolated all the proteins of a cell. And I said, well, I want to flag this protein insulin that came from this mouse. And so I have an antibody that will bind specifically to insulin. And let's say that I've dyed my antibody so it gives a brilliant green color. And I take my mixture of proteins right here, and I dump my antibody into it, and I see green, and I go, wow. I can't tell anything. Why? Well, the green came from the antibody. I don't know if anything is bound. It's all stuck in this tube. So if I want to be able to use this in a meaningful way, I have to separate the proteins and then use the antibody to bind to those separated proteins, because then I can see, is it binding to everything that's in there? Is it binding to one thing that's in there? Or is it binding to nothing that's in there? Right? So that's what Western blotting is all about. It involves running, first of all, an SDS page gel. SDS page, you recall, allows us to separate the proteins on the basis of their size. And after we've run that, we transfer, by using electricity actually, those proteins that are in the gel slab, we transfer them onto a membrane. And we set the membrane up so that it literally binds to those proteins and won't let them go. The covalent bonds are actually formed. Okay? So the proteins that were on the gel are now sitting there on that membrane, and they're not going anywhere. They're in exactly the same position they were on the membrane. I then take that membrane and I put it into, most commonly in the laboratory, we actually use Glad bags. Okay? You take a bag, all right, and you have a buffer, and in that buffer you place that antibody. You let it mix for a period of time, and then you wash off the excess. So you take it and you dump it in a few solutions. Now, if you have binding, the washing isn't going to remove the interaction between the antibody and the protein, whereas if you don't have binding, all the excess antibody is going to get removed. So then you can look at this and say, oh, I did have that protein there, here's how much I had, and it's of this size. And that's what Western blotting tells us. Make sense? Good. Yes?
When the proteins are covalently bound to the membrane, doesn't that change their shape? Wouldn't that denature them? Or?
That's a really good question. Okay. One of the questions that people commonly ask is, wait a minute, SDS, isn't that a detergent? Isn't that going to denature the proteins? And wasn't that the idea of this whole process in the first place? And the answer is, it can be a factor. It can be a factor. So just because you see nothing doesn't always mean that the protein isn't there. Maybe the antibody doesn't recognize the denatured form. Maybe there's other things blocking it from getting there and binding to that. So you're exactly right. That's possible. Several good questions tonight. But I think we are at an end. So if you want to call and talk to me, send me an email. I won't give my number out to the YouTube crowd. And if not, I'll see you in class at 9:30. Study hard.
What's the oddest phone call you've got?
Nothing too odd. But I've had people call up and pronounce that, hey, I saw your thing on YouTube. Do you really take questions? Okay, sure.
What calls you got were about the class?
Yeah. Oh, yeah. And I answer their questions, too. Yeah.
Is there a BB 452 also?
There's not. There used to be, but there's not. No.
Okay.
What is it about the angiogenin?
Angiogenin?
It's the one that lets the blood vessels go around the road. So we're blocking it. So